Hi, in today's series of videos, we'll be talking about cytoskeletal proteins and the interaction of cells with the extracellular matrix. The cytoskeletal proteins are structural elements of the cell that give strength and rigidity to its shape and also promoting cell mobility. There are three main types of cytoskeletal elements. In this module, we'll talk about two of them, intermediate filaments and microtubules, saving actin filaments for another video. We'll conclude with a third video talking about interactions between cells and the extracellular matrix. This slide is mostly for looking at this picture, which I think gives an excellent visual introduction to cytoskeletal proteins. In green, we can see the microtubules moving radially like spokes from the center of the cell, and then we can see a band of actin around the periphery that's helping to project these filaments, which are a component of generating cell movement. In blue, for reference, you see the DNA in the nucleus. In addition to cell movement, macroscopic movement, we'll talk about how the different cytoskeletal elements allow for organelles to move within a cell, and proteins, vesicles, and other subcellular elements. The three cytoskeletal filament types are intermediate filaments. These have 10 nanometer cross sections. They are purely structural. They provide strength, mechanical strength to the cell. Microtubules are both structural and dynamic. They have a large cross-sectional size, 25 nanometers. They function primarily as the cell's highway or roads. Organelles and proteins that need to be transported to different parts of the cell hitch on for a ride along microtubules. Finally, there are actin filaments, which have the smallest diameter. They are contractile. They interact with myosin in muscles. They have both structural and dynamic functions and play a particular role in whole cell locomotion. We'll start by focusing on the intermediate filaments, and the first thing to notice is their schematized arrangement within a cell. They tend to run around the periphery of a cell. You can see them at focal points where there's interactions between cells. There is also a network of intermediate filaments inside the nucleus, giving strength and shape to the nucleus. And one thing to note, that while they tend to be at the periphery, they're not extending up here into the cilia or fimbria-like projections of the cell. That will be where actin networks are predominant. This is an electron micrograph, and here's a schematic. We'll see more about the structure on the next slide. Intermediate filaments are the strongest of all the cytoskeletal proteins. They have the highest tensile strength, and their function again is to resist mechanical stresses. The basic structure of intermediate filaments is that they form parallel dimers and then anti-parallel tetramers, so a pair of dimers, the amino end here, the amino end here. Finally, you get 8 tetramers together that get twisted into this rope-like element. Structural features that add to the strength is the offsetting of the units, such that there can be a great amount of overlap between one of the tetramers and a next set that would start here. As we'll see later, there are a wide variety of proteins that can form these intermediate filaments. They tend to all have homology in the central region, but it's these end pieces that vary between the different types of intermediate filaments that can form. This slide shows an immunofluorescence photograph of epithelial cells plated on a petri dish, and it emphasizes the fact that these intermediate filaments are not only giving structure to the cells, but also connections between the cells, so allowing this sheet of cells to withstand mechanical stresses at places called desmosomes, which we'll talk more about in a later lecture. Another aspect of the structural features that this slide sort of makes apparent is if a cell experiences a mechanical stress on one side, that this network can help distribute and dissipate the forces so that impacts to any particular region of the cell will be minimized. Intermediate filaments are a key component of epithelial cells, and epithelial cells are the types of cells that are present at the boundaries of your body with the outside world. In the skin, obviously, but also on the inside of your body, at the digestive tract where the body meets the food, in your lungs where your body meets the oxygen, and these tend to be regions where there are a lot of mechanical stresses. As has been emphasized, there's lots of connections between epithelial cells in addition to fortifying the intact cell structure, and there are actually diseases that occur where these connections between cells are impaired, such as epidermolysis bullosa, in which just simple mechanical stresses, rubbing of skin, can cause large blisters where skin is just peeling off in sheets. As we mentioned, there are different types of proteins that are involved in different tissues. You don't need to remember any of these names, but you've heard of them, many of them anyway. Keratins are the intermediate filaments in epithelial cells and hair. Vimentin is a primary protein that forms intermediate filament in connective tissue and muscles. Various neurofilaments in nerve cells. These can play a role in the disease processes of ALS. And then all cells have nuclear lamins, which is a meshwork of intermediate filaments giving structure. Intermediate filaments are connected to each other and also microtubules by a protein called plectin. This is going to form cross-links, which greatly strengthens the cell's ability to withstand forces. This slide details the nuclear lamina, and here we can see the intermediate filaments that reside just on the inside of the nuclear envelope, the double membrane structure. This is a mesh-like configuration of these proteins, and they also serve as scaffolding. There are also connection proteins which connect DNA and anchor parts of chromosomes to the nuclear lamina, providing structure and shape of the chromosomes as well. One minor point is these proteins are called lamins, and you should not confuse them with laminin, structural protein that exists outside the cell. So these are nuclear lamins. All right, so this will be a good point to pause, and we'll resume talking about microtubules. Thanks for listening.